Hepatitis E virus research has been hampered by the lack of an efficient cell culture system. Our techniques overcome these limitations, paving the way to drug design and vaccine development.
With this protocol, we are able to produce infectious high-titer virus stocks of both non-enveloped and enveloped HEV particles, facilitating the infection of a variety of cells. Execution of this protocol requires access to a BSL-2 facility and experience with basic cell culture techniques.
To linearize the plasmid DNA, mix 10 micrograms of the template DNA with 10 microliters of buffer and 2 microliters of the Micrococcus luteus restriction enzyme, and adjust the final volume to 100 microliters with water. Then incubate the solution for 1 hour at 37 degrees Celsius and confirm linearization of the plasmid by agarose gel electrophoresis according to standard protocols. After plasmid DNA isolation and linearization, the success of the linearization can be verified by comparing the non-digested plasmid DNA to the digested plasmid DNA by gel electrophoresis. For in vitro transcription of the purified full-length hepatitis E virus DNA, mix 2 micrograms of the linearized DNA template with the appropriate reagents and use nuclease-free water to bring the final volume of the solution to 100 microliters. After thorough mixing, incubate the solution for 2 hours at 37 degrees Celsius. After 2 hours, add an additional 2 microliters of T7 RNA polymerase to the tube. Then mix the reaction